Hi, welcome back. Okay, so now that we are clear what proteins are made out of and what codons are responsible for which amino acid, we are ready to talk about the process of translation. Again, for eukaryotes, this takes place in the cytoplasm using ribosomes compared with transcription in the nucleus using RNA polymerase. But the directional movement of translation doesn't change from transcription or DNA replication. So again, here I have noted that translation takes place in the cytoplasm on ribosome, but again, it always goes from 5' to 3' direction. So this is your mRNA molecule that has traveled to the cytoplasm and hops on the ribosome, and it always hops on near the 5' end, and it will move down the mRNA towards the 3' end, and then you will have a resulting polypeptide chain. The ribosome will always attach near there at the 5' end, okay? And it will always go towards the 3' end. And synthesis always begins at the amino end of the protein, and then it will be elongated by adding new amino acids to the carboxyl end. So if this was your amino acid 1 right here, your very first amino acid, that's why it has an N at the end, signifying that what's sticking out of there is your amino group, central carbon, and then your carboxyl group that forms your peptide bond with your next amino group of the next amino acid, central carbon, carboxyl, C of the carboxyl group, hydrogen, R group, okay? And again, this is hydrogen, R group. Then this is your peptide bond. Okay, so that's why the amino group always starts your polypeptide chain, and it just keeps building a new peptide bond at the carboxyl group of the amino acid. And finally, the last amino acid would have a carboxyl end, COO minus, as the last amino acid there. And this N represents your NH3 group, okay, which is the amino group end of the polypeptide chain. Okay, so let me erase that so it doesn't messes up the other next segment. Okay, so there are 4 stages of translation. We still have initiation, elongation, and termination, just like DNA replication and transcription, but we have one additional preliminary step before initiation for translation. And that is the first stage called tRNA charging. So this is when your amino acid gets attached to a specific tRNA molecule, because the amino acid can't just travel and somehow binds to the mRNA molecule on its own and it starts connecting with each other to form your peptide bonds. You need to have something that brings the amino acid towards your mRNA molecule. In this case, that's why it's called transfer RNA for tRNA, because it actually transfers the amino acid to your messenger RNA. Remember, we talked about how the secondary structure of tRNA is that clover leaf-like structure, and that in the amino acid is attached to the 3' end of the tRNA. So this is that clover leaf structure right here. Here's that 3' end, and the amino acid, it is attached to the 3' end of the tRNA molecule. It is the carboxyl end of amino acids that gets attached, leaving a free amino group for forming that peptide bond in the next incoming amino acid. So this is that carboxyl group of your amino acid. This is your amino group. There's your R group. So this carboxyl group is what forms a temporary bond to your tRNA molecule via adenine. There's an adenine phosphate group and the rest of your tRNA molecule. So this bond is formed for attaching the amino acid to your tRNA molecule. The attachment of amino acid to the tRNA is catalyzed by an enzyme called amino acyl tRNA synthetase. It's a long name, 3-word name, but in each amino acyl tRNA synthetase is specific for one amino acid. Hence, that is why there are 20 of these enzymes for the 20 different amino acid. One specific enzyme adds one specific amino acid. If we are to add, for example, alanine to this tRNA, there's that one specific amino acyl tRNA synthetase for that alanine amino acid. And then if we're going to add proline to the next as the next amino acid to the tRNA, then another different amino acyl tRNA synthetase specified in adding proline would be used. So there are 20 of these enzymes, again, for the 20 amino acids. This process, again, requires ATP to power the reaction. Now, because there are multiple codons for a specific amino acid, is not just one anticodon in a tRNA per amino acyl tRNA synthetase. There can be multiple combination of different anticodon on the tRNA that uses the same amino acyl tRNA synthetase enzyme for attachment of the same amino acid. Remember, in your clover leaf-like structure of the tRNA, the bottom here is where you have your anticodon. So not every anticodon gets a specific enzyme for adding that amino acid. What if there were 6 of these anticodons, for example, that adds leucine? Then all 6 of those tRNA will use the same amino acyl tRNA synthetase for adding leucine. There are actually 30 to 50 different tRNAs available, but again, there are only 20 amino acids, which means only 20 of these synthetase enzyme. So just like how we have synonymous codons, we also have what's called iso-accepting tRNA, such that one amino acid, meaning that multiple different tRNAs, which means they can have different anticodons, can encode for the same amino acid. Okay, doesn't mean that different tRNA will have different amino acyl synthetase. It just means that different tRNAs can encode for the same amino acid using the same amino acyl tRNA synthetase. Okay, just be clear with those two differences. Now that we have a bunch of charged tRNAs ready with their amino acid, we can begin translating the mRNA molecule into a protein. Now, initiation is when the mRNA molecule has initiation requires your mRNA molecule, your small and large subunit of your ribosomes, your initiation factors, an initiator tRNA, and some energy provider, which is specifically GTP for translation. These are what's required to form what's called the initiation complex. So first you have the 30S, okay? 30S means the small subunits of the ribosome. The 30S attaches to the mRNA molecule here, okay? Then an initiator tRNA right here. Initiator tRNA binds in that specific region on the mRNA molecule through base pairing between the codon of the mRNA and anticodon of the tRNA. This is always at what's called the start codon. So remember, I was talking about this when we were going over different genetic codes or codons for different amino acids, and I said start codon is one of those that you have to know the sequence of, which is AUG. Okay, it always begins with AUG when we talk about translation initiation stage. It doesn't start, it never starts with another codon. It always starts with AUG, which corresponds to the amino acid methionine. And hence that is why you have MET right here attached to it. Okay, so AUG encodes for methionine, okay, or MET amino acid. So every protein pretty much out there that you know of will begin with the amino acid methionine. Now, once you have the initiator tRNA, the mRNA molecule, and then your small subunits of the ribosome, this is now called the 30S initiation complex. Then the large subunit of the ribosome, which is known as the 50S molecule, joins the group and make your 70S initiation complex, and now we are ready for the next stage in translation. Now, the initiation step in bacteria is again a little different from eukaryotes. The mRNA in bacteria contains a very specific sequence called the Shine-Dalgarno sequence. Shine-Dalgarno sequence. This is absent in eukaryotes, only in bacterial cells. They allow for that binding of the small subunits of the ribosome, which is your 30S. In order for 30S in bacteria to bind to their specific mRNA sequence, they have to have this yellow section called the Shine-Dalgarno sequence, whereas for eukaryotes, we don't need that. You can see that in this figure here, right? In this figure here, you have lots of IFs. All those IFs you're seeing is what we call the initiation factors. Initiation factor, that's what it stands for. Now, I'm not asking you to memorize which initiation factor does what. They are all critical for formation of this particular initiation complex, okay, initiation complex, but it is not crucial to remember all their different IF1, IF2, IF3, what exactly do they do? Now, bacteria have exactly 3 initiation factors. And again, you don't have to know the details of each. And lastly, their initiator tRNA is something special where their methionine is slightly modified, and it is called the N-formal methionine. So that's why what you're seeing here is actually your bacterial initiation, because it includes all those parts, the Shine-Dalgarno sequence, and also their initiator protein right there actually says FMET, which is for this N-formal methionine. Now, this again only exists in bacteria. In human eukaryotes, we do not have such thing as FMET. There's no distinguish between internal methionine versus your initiator methionine. For bacteria, it's slightly different. So for eukaryotes, there are again no such Shine-Dalgarno sequence, but remember, we do have mRNA processing steps in eukaryotes. And one of them that we talked about last chapter was the 5' cap. And we said that 5' cap actually helps with the initiation process of translation. So here it is. The 5' cap is what the small subunit of the ribosome will recognize and bind there. So small subunit is not looking for a shine Delgarno sequence. For us, it is looking for the 5' cap. Initiation factors and initiator tRNA has, which now for your eukaryotes just have normal methionine. It does not have F-mets. Okay, so their tRNA, if you want me to draw that, okay, then it would just have methionine at the end, no F-mets for eukaryotes. So then all those then will come together as your initiation complex. And actually, once that initiation complex is formed, they will sort of scan and run through your mRNA sequence looking for that AUG sequence. So it just scans along the beginning of your 5' end of the mRNA molecule and then looks for that very first AUG codon, and that's where translation starts. That start codon. And that's how we know from which reading frame on are we going to read your codons. Because technically speaking, if you think about it, you have your mRNA. Let me go grab a blank board. You have your mRNA molecule, 5' N, 3' N. You have your 5' UTR region, 3' UTR region. And then you have, maybe random sequence, and then something like that. Okay. So technically speaking, if your small subunit of the ribosome can recognize any of these codons, then we don't know where the code reading frame starts. What if we started here? What if we started here? Okay, so depending which nucleotide your reading frame, your very first 3-letter codon starts, that's what determines where your translation will start. Okay, so now that we know translation must start at AUG, okay? Ooh, okay, let me go back to pen. Okay, so CC and AUG. So this sequence here is actually meaningless because what matters is you have your large subunit of ribosome, small subunit of the ribosome, your initiator tRNA molecule with your methionine attached. It's going to go ahead and scan and move in that direction of your mRNA molecule towards a 3' N until it finds that one AUG sequence. So that's your start of the translation, where your start codon is where then we'll begin. So from here on out, then you start reading every 3 nucleotides after your AUG. So translation doesn't necessarily start at the very first letter after your UTR region. It has to look for your start codon in order to continue the process of translation further down the road. Okay? All right. So now the other thing different for eukaryotes is that there are at least 12 different initiation factors. Notice the at least, which means there can be more in different types of eukaryotes. There's just too many complexity there. But again, this tells us how eukaryotes is a lot more complicated than prokaryotes. Everything, even with DNA polymerase, we said there's only polymerase 1, polymerase 3 for prokaryotes for DNA replication. But for eukaryotes, there's actually 12 or more, or was it 14? But there was a lot of DNA polymerase, and I said you don't have to worry about those, okay? So this is why scientists in the beginning back then chose to study prokaryotic mechanisms first before eukaryotes, because they're a lot simpler to understand the basic foundation of these processes first. And even when we're talking about basic, this is not simple. This is still many steps involving a lot of stuff, but it's just much simpler already compared to eukaryotic cells. All right. So the last thing that differs in eukaryotes in the initiation step is that there are proteins attached to both the 5' cap of your mRNA molecule, and then there are proteins that is attached to the 3' poly A tail of the mRNA molecule. The proteins that are attached to these regions, for example, this yellow area right here and this red area right here, these two proteins will actually interact with each other, and they like to attract each other. So this interaction between proteins that are coming from the poly A tail and proteins that are coming from the 5' cap actually cause the mRNA molecule to end up bending on itself, forming a closed circular loop like this right here. Okay, so that's why if you watch videos online with translation, you might see the depiction of circular mRNA with the ribosome reading throughout that circle. Is because for eukaryotes, this is what happens between the interaction of the 5' cap and the poly A tail. So this structure itself actually assists with proper binding of your ribosome to your mRNA molecule to ensure that it's not erroneously binding somewhere else, or is not fitting right, and then translation completely ceases. So this further confirms that right position of the ribosome. Okay, so the third stage of translation is then elongation. This is when new amino acids are added one by one to the growing polypeptide chain. It requires your 70S initiation complex that we just formed in the initiation step, your tRNAs that are charged with amino acids, your elongation factors. Notice that it's easy to remember these names, especially for these so-called factors, because they kind of name after the stage itself. So initiation stage requires initiation factors, elongation stage requires elongation factors, and of course all of these translation stages requires your energy molecule GTP. Okay, elongation then goes in a cycle. Okay, I rather use this figure from my 1406 class than your genetics textbook, because I think the genetics textbook made it even, looks a lot more complicated than it needs to be. Elongation is very simple. It's just 3 steps that goes in cycle over and over and over again, involving 3 sites on the large subunit of the ribosome, your A, P, and E site. So on the ribosome right here, for example, this is your ribosome. You have your A site, P site, and your E site. Okay, each of these is where your tRNA does something different. Okay, it will be catalyzed by the ribosome to do something different. So the amino ACOA site is where the next specified amino acid with its tRNA will arrive and bind. Okay, so, and I like to tell my 1406 students the easy way to remember this is A site for arrival of the next amino acid. Okay. And then the peptidyl P site is where a peptide bond is formed between the amino acid in the P site and the amino acid that's in the A site. Okay, so the amino acid is cleaved off of its tRNA molecule in the P site, so the strands, the whole polypeptide chain kind of hops over to the tRNA in the A site. So here is depicting that. You have your ribosome, right here I'm talking about. You have your tRNA in the P site with already a growing chain of amino acid. Here's your incoming tRNA with a new amino acid that has anti-codon sequence that is complementary to this next codon on the mRNA. So the tRNA comes in, binds at the A site, and then what catalyzes the next reaction is the movement of this entire polypeptide chain that's growing to the next amino acid in the A site. So that's moving, cutting this bond away, moving this chain to the A site, and that's why now you have the chain attached to the A site. Your P site is now empty. Okay, your tRNA in the P site is now empty. It no longer has amino acid attached to it, so then it no longer is needed. So that's when we're moving on to the next site. So how it moves on to the next site, the tRNA actually doesn't know to hop over to the next E site. The ribosome is what's going to do your translocation. It will translocate to the next codon over, which is here, your next codon over. As the ribosome moves, your mRNA, tRNA stays in place, and hence your P site tRNA then gets translocated to the next site over, which is your E site. The E site is called your exit site. So this is where your free tRNA will then be released back into the cytoplasm to be charged again by your amino acyl tRNA synthetase and attach another molecule of amino acid, and that particular charged tRNA can then be reused again for adding the same amino acid that's next in the sequence. And hence, the ribosome actually cycles through these 3 sites, starting with P as your central tRNA. Then you add your tRNA to the A site. Amino acid chain hops over to A site. The entire ribosome moves on and translocate this way, further that way. So then you can move this tRNA to the E site, and then your A tRNA move over to the P site. And hence your tRNA in the E site exits away. Your P site tRNA now will start the cycle all over again with your next set of codon. So it keeps repeating and repeating and repeating itself until you reach the end of the mRNA, not the very end, but the stop codon, and I'll talk about that in a little bit. All right, so, and I'll show a video that, show this, all these steps more animated style, so it's easy to visualize. Okay, in the beginning, when the initiator tRNA binds to mRNA molecule with the small subunit of the ribosome, it actually puts that start codon tRNA in the P site. So you're talking, if you go back to the previous slide here where we have your complex, your 30S initiation complex. So you're like, well, there's that initiation complex, your mRNA, okay, your initiator tRNA, your mRNA. So what site is this? Is this going to be in the A site or P site or E site? Well, it turns out that this initiator tRNA is always going to be over the P site, okay, to start the whole process, because technically speaking, your next amino acid should be arriving at the A site instead, okay? So your P site is where you start, and then the A site starts your next amino acid, okay? Now, one thing to add about elongation step here is that mRNA and tRNA pair according to their complementary sequence in that anti-parallel fashion, just like the 2 strands in a DNA molecule. So you have your 5' to 3' here on the mRNA molecule. And the anticodon would then the opposite, where the 3' is on this side and the 5' end is on that side. So that's the anticodon part of the tRNA. So see how these two are in anti-parallel fashion, just like your 2 DNA strands. So the first and second nucleotide on the codon always binds by complementary pairs, which means A always binds to U, G always binds to C for the first two nucleotide position on the codon. But the third position is where things can get a little bit more flexible. It does not need to be exactly the complementary pair. This third pairing can be more relaxed, where G on the tRNA's anticodon, as you can see here, G in that third position, can actually pair with a U or a C. So G, technically speaking, should pair with a C, okay, by complementary sequence. But you can see here that it actually can pair with a U. So this idea of having more flexibility on the third nucleotide of the codon is known as wobble, which means it can wobble a little bit around, move around a little bit. So it can wobble and move around a little bit in that third binding complementary pairs. It doesn't have to be exact. So this shows that slight flexibility we can have in the third nucleotide position of a codon, which explains why some tRNAs can pair with more than one codon on the mRNA molecule, or why synonymous codons can specify the same amino acid due to its flexibility. Remember, in the first part of the lecture where I talked about the synonymous codons, we said that usually with synonymous codons, they are differ in the third position of the nucleotide in the codon sequence. And this wobble idea is what explains for that, because the third position allows for more flexibility in that sense. Okay? All right. So the last stage of translation is called termination. This is when protein synthesis, of course, have reached the end of the amino acid sequence, not the very end of the mRNA molecule, because you still have the 3' UTR region, but it will stop when it senses any of the 3 stop codon, and then the whole initiation complex will dissociate. Remember that we said these 3 stop codons are your UAA, UAG, and UGA, and you should know the sequence of these 3 stop codons. When a ribosome has reached any of these 3 codons, no new tRNA will pair and arrive at the A site of the ribosome, which means there will be no amino acid incorporated on the side when your ribosome has reached your stop codon. No new amino acid will be incorporated once a stop codon has been reached. So instead, a release factor is what binds to that A site. It's not tRNA anymore when we have reached the stop codon. This particular release factor protein will promote the cleavage of your polypeptide chain from a tRNA P site. So let me erase what I had earlier here. Okay, here. So it doesn't get in my way. All right, so now you have your, this is illustrating where your ribosome has reached that one stop codon, UAG. So notice that no tRNA is bound there. Instead, something called a release factor is what's going to bind to that A site of the ribosome, which then causes the cleavage of this particular bond between your tRNA molecule and your string of amino acid. It cleaves and cut that off, and out goes your polypeptide chain, and hence your tRNA will then be dissociated as well. Your tRNA will actually moves to the E site first, and then be exit out of your large subunit of the ribosome back into the cytoplasm. Your mRNA molecule itself will be released from the ribosome, and then the entire ribosome will also dissociate back into their individual large subunit 50S, and then your small subunit 30S. Don't ask me why 50S and 30S don't make 80S initiation complex. That's the way they have named these complexes. So 50S plus 30S, somehow you get 70S initiation complex. I'm sure there's a reason to that for whoever named that. Okay. So remember that the component of an mRNA molecule has a 3' UTR region. Remember, that's your untranslated region, which means there's no encoding of proteins or amino acid within that region, and the stop codon is not at the very end of the mRNA molecule. It is actually, before your 3' UTR region, and usually there are some nucleotides left over. All right, so this is a good summary slide for the 4 stages of translation, where here you have your first step, tRNA charging, where then, remember, it involves your amino, ACO, tRNA, synthetase. Excuse me for my handwriting. It's hard to write while the iPad is upright. Okay, synthetase. So this is the enzyme that adds that particular amino acid to your tRNA molecule. Now, during initiation step is where you have your mRNA molecule right there, and then you have your initiator tRNA, and then your 30S small subunit of the ribosome, and then you have your 50S large subunit of the ribosome, and of course many other initiation factors that come into play of putting this 70S initiation complex together. Okay, so that's what we get at the end of initiation. Then you have elongation, which cycles through your 3 sites on the ribosome. EPA, A for amino, ACO, which is then you're having that arrival of your charged TNA to that site. A for arrival, if you want to remember that. And then your P site is peptidal site, which means is for in charge of making that peptide bond between your growing chain of amino acid to the newly incorporated amino acid. You're forming a peptide bond between these two, and then the tRNA that's been cleaved off of will then enter your E site, and then out goes it back into the cytoplasm for recycling. It will become charged again, where then it can come back here to start the whole process again. And now this particular ribosome will continue moving downwards towards your stop codon, UAG. And don't forget that you have your start codon right here, AUG, is where for bacterial cell, it will bind right there according to your Shine-Delgarno sequence. But for eukaryotes, it will actually scan and look for that first AUG. And then once you have reached your stop codon, this is when termination stage of translation begins. Your release factor binds instead of a tRNA molecule at the A site, which then causes the cleavage of your polypeptide chain from this tRNA. This tRNA then hops over to your E site, out goes your tRNA, and back into the cytoplasm and recharged again. Okay, and hence and then the rest of your mRNA complex, your mRNA molecule, your large subunit, small subunit, all dissociates apart, and you have your completed polypeptide chain that will either fold onto itself due to those interactions we were talking about in the 4 levels of molecular structure of the protein, or sometimes other proteins will require the help of other proteins that will help it fold into this final shape. And I'll talk about that in a little bit, okay? So I will remember to upload an animation video to eCampus after my lecture video that will depict the process of translation in bacteria. Now, this is a table summarizing the different components of each translation process. Again, you don't have to worry about these different initiation factor and elongation factor. You don't have to memorize these names. Only those parts we actually talk about within this lecture video is what you need to know for the exam. For example, amino acid, tRNA, amino acid tRNA synthetase, mRNA, SMak tRNA. Remember, that's only specific for prokaryotes. Bacteria, 30S subunit, 50S subunit, and then those initiation complex that's made, your charged tRNA, GTP, and your release factors, okay, but not the names. But at least knows that you do involve initiation factor and you do involve elongation factor, but you don't have to know the specifics of which elongation factor does what, which initiation factor does what, okay? All right. The last section of the chapter talks about two interesting that happens during or after translation. The first thing is that we have seen as one ribosome to one mRNA molecule, which is what we've been depicting in our little cartoon earlier in all those mechanisms, is actually too simple of a model. What actually happens in both prokaryotic and eukaryotic cells is that multiple ribosomes can attach to one single mRNA molecule at the same time to simultaneously make multiple copies of the same protein at the same time. So this really speeds up the process of translation, and we don't need to transcribe 100 copies of mRNA molecules to make the same 100 molecules of the final protein product. Okay, just like how we have one recipe for making a carrot cake, okay, but we need to make 100 carrot cake for a buffet or a party, but that doesn't mean we have to make 100 copies of this recipe. We already have that one recipe. We're just repeatedly going through that same recipe again. And if you have more workers, like your ribosomes are your workers, then you have multiple people baking that cake at the same time, reading that same recipe you started off with for that carrot cake. Okay, so that's the same idea here. We don't need to make multiple mRNA transcript for the same final protein product. It is just not efficient. Our cells always like to figure out a way to do things efficiently with less starting material, less energy, and produces a lot of product. Okay, so how scientists discovered this idea again is by the evidence of a visual image on the electron microscope. That's why electron microscope, the discovery of that machine changed a lot about these biological concepts, because then we can see really what happens or have a clue as to really what happens in a cell. They saw that in the string of mRNA molecules, there were bead-like structure all around the mRNA strings. So these are your ribosomes, turns out. In an mRNA molecule, they can see that there are multiple ribosomes all around the entire transcript, which means multiple ribosomes can translate the mRNA molecule at the same time. And this is called polyribosomes, this mRNA, not the ribosome itself. We're referring to this mRNA molecule with multiple ribosomes attached to it called polyribosomes, okay? For prokaryotes, transcription, remember, and translation actually happen simultaneously in the cytoplasm. They can happen at the same time because it's taking the same three-dimensional space. So we can observe polyribosomes while transcription is still taking place at the 3' end. As you can see here, transcription is still happening, but at the same time as the mRNA molecules is exiting all of that transcription bubble, you will see all these ribosomes already attaching to your mRNA molecule. Already can't even wait till the mRNA finish to get reading on the mRNA molecule and start chunking out that protein that's necessary for the process. Now, for eukaryotes, transcription happens in two different locations, okay? Transcription happens within the nucleus, translation happens in the cytoplasm. So we will never see this happening in eukaryotes. This is only for prokaryotes, but in the cytoplasm, there can be polyribosomes in eukaryotes as well. So after translation, like I was talking about, some proteins can automatically fold on itself in that tertiary structure to form that functional three-dimensional shape. Others may actually require some help. And these helper proteins in assisting that three-dimensional folding, it's called molecular chaperones. They will guide and assist in proteins folding into their final 3D shape, just like depicted in this picture. Here's your ribosome, here's your mRNA molecule, and here's that polypeptide chain coming out of the ribosome. After it's done and released from the ribosome, translation is completed, then it can actually goes through this one of these chaperones. And after it comes out of the chaperone, now it is in that nice folded three-dimensional shape of the protein instead of just this simple thing here that is non-functional yet. Okay, so sometimes some protein requires chaperone proteins to help. In addition, sometimes proteins will have what's called post-translational modifications, meaning after translation, some modification, chemically speaking, can happen on an amino acid. That's why Golgi apparatus in a cell does modification and repackaging of the proteins before they are sent to their final destination. As you have learned in your cell organelle, what are they responsible of? Okay, so Golgi apparatus was one of those that says you're repackaging your protein. So this repackaging and modifying is part of that is these post-translational modification, and it can include a whole bunch of things. The protein sometimes is cleaved or trimmed to a shorter size. They can attach carbohydrates to it. So remember, glycoproteins that sits within your plasma membrane have these little sticks coming out of it for recognition purposes. This is a carbohydrate molecule. So proteins like this needs the attachment of these carbohydrates to make glycoprotein. Other things like phosphate group, carboxyl group, methyl groups can also be added to individual amino acids to change in their shape or particular function. And hence, this then concludes chapter 11. Again, a short and straightforward chapter. It's just a lot of explanation to go through. Now,